Hello and welcome to Biorules video lessons. Today we are going to be talking about movement of particles across a membrane. So in biology, physics and chemistry, particles they move, and there is one special kind of movement called diffusion that is the movement of particles depending on the concentration. So as an example here we have a crystal of potassium permanganate, where so I have a very high concentration of this potassium permanganate molecule in water. This particle, it diffuses in water, so it's soluble in water, so it's going to be able to get diffused over there. So what's going to be happening? This here is what usually we'll see. It goes in moment A to start moving away from the high concentration, so A, B, C, D, so the tendency is for this chemical to spread around it to, after a while, to be evenly distributed. So basically what has just happened? To show this, let's illustrate each particle here as one little ball. So we have here this potassium permanganate or any other dissolved chemical in water, and here is they are highly concentrated in the top left corner of this bottle. So what's going to be happening is that particles, when they are dissolved, they do something called Brownian movement. They move kind of randomly, bouncing everywhere, and as a tendency they tend to spread around, and to move from where they are highly concentrated, to bounce around and randomly in the chemical. So these molecules are going to be bouncing there in the chemical, and the tendency will be for them to spread from where they have a very high concentration to where they have a lower concentration. So here what we have is that these particles, they are filling up the environment, and the impression that we have is that they moved from high to low concentration. Actually, they keep moving. Even when they are evenly spread around, they keep moving, but now we don't see them moving anymore because they are not changing concentrations around this environment that they are. Okay, so diffusion. We have there, if I put a dye molecule, the tendency of them is to be spreading around looking for the equilibrium. It's a natural tendency. They spread around from high to low concentration. They actually spread around from high to where they don't have any chemical, or basically everywhere, and they end up equalizing concentration. So it looks like they're moving from high to low concentration. Okay. Examples of diffusion. One very good example is when you dissolve gases, carbon dioxide, water vapor, or oxygen. They might pass through the membrane quite easily. Water is a special case. I'll mention that in a while. Another example is when some chemicals don't manage to pass through the membrane and they have to pass through channels, pores going around. For example, food particles, when we eat, we absorb food in our intestine, and that usually happens by diffusion. So there are a few different cases of diffusion. One of them is called simple diffusion, because molecules simply diffuse through the membrane. Gases and other molecules that are not hydrophobic, they don't care for the membrane itself, they just pass through. They are not blocked by the hydrophobic part, so they simply squeeze there. They pass directly through the membrane, no need for special proteins or nothing. So typical example, gases, oxygen gas, carbon dioxide gas, nitrogen gas, they just pass through. What about water? I said that water vapor was one example. So what happened to water? Not only water vapor, but to water itself. Most water molecules, they don't manage to pass through the phospholipids. If they try, if they manage to start entering there, they get squeezed out and they get away from here, and they end up bouncing out of the membrane, and it doesn't manage to cross. However, when the water molecule enter the phospholipid bilayer and get to the hydrophobic part, they are expelled to somewhere. They basically they get kicked, and most often they are kicked out, which is the closest place for them to go. But sometimes they are kicked to the side, and they are kicked again, and go away, and they might end up going away, bouncing around. They might get out in the other side. This is not very common, but because of the huge number of water molecules that you have in both places, some water molecules end up being expelled by the phospholipid bilayer like that, and they end up actually being able to cross. It's not many of them, it's a few of them, but because we have so many water molecules, some of this end up happening and they go to the other side. Okay? Some chemicals, they don't manage to cross through the phospholipid layer itself because they are charged, because they might be polar, they might be hydrophilic, they might be sometimes a little bit too big, so they don't manage to squeeze themselves through the membrane. They need a transporter. These transporters are called channels. On the left we have a non-specific channel, so it transports any positive ion, so it can be a sodium, it can be a potassium, it can be whatever. And on the right we have a specific transporter, in this case here a glucose transporter, which transports glucose. It's not going to be transporting fructose, it's not going to be transporting any other monosaccharides, it will transport glucose only. So channels are very good because sometimes they let molecules to pass through. Okay? They allow molecules that are not capable to squeeze through the membrane itself, they still pass through the membrane because they're passing through protein channels in the membrane. So we call that also crossing the membrane. And those are what we are going to be calling passive transport. So on the very left over there we have simple diffusion, we have the facilitated diffusions here in the middle, that the ones that use a channel. We even have the water on the right, that can be simple diffusion or through a water channel called aquaporing. All of this, they happen naturally. There is no need for energy, there is no need for push. So this is the definition of passive transport, because simply go to the other side, they go from high to low concentration, they go in favor of the concentration gradient, so they don't need any energy to be put for them to cross through the other side. Simple diffusion, facilitated diffusion, examples of diffusion, passive transport through the membrane. Okay? What if a molecule wants to go from low to high concentration? What happens if you have something like this and the molecule wants to go that way? What will happen if you have a channel is that this channel might allow this molecule to go that way, but it will also allow the other molecules to come this way, and the result is going to be much more of a movement in favor of the gradient than against. So channels are not good for movement against the concentration gradient from low to high, so this is not going to happen, but sometimes cells want to push chemicals against the concentration gradient. So what happens in that case is that there are special proteins called pumps that they do active transport. They actually accept energy, usually in the way of ATP, adenosine triphosphate, that change its shape, and when it changes shape, it allows particles to be squeezed from one side to the other. So this here makes proteins to go from low to high concentration in what we're going to be calling an active transport, and that's actually this used a protein called a pump. Okay, so pumps are very useful to make chemicals to move against the concentration gradient. So here you have examples. On the left you have passive transport, simple diffusion with the purple balls, and facilitated diffusion with the red triangles and the blue balls. The simple don't need a protein, the facilitator needs the proteins to pass to the other side. And active transport, you have a classic example of active transport, which is the sodium-potassium pump. It pumps 3 sodiums to one side, while it pumps 2 sodiums to the other side with the use of 1 ATP. 1 ATP, 3 sodiums to one side, 2 potassium to the side, a classic pump that we use a lot, especially in our brain. We are going to be using a lot of these pumps. Okay? One thing that you should be able to do is to compare active and passive transport through the membrane. So for that, I'm going to be building a table, because tables are nice for us to be doing comparisons. So what about the concentration gradients? Passive transport follows or go in favor of the concentration gradient, and active transport goes or can go against the concentration gradient. It's the same thing if we say it's going from where to where. So in the passive transport, it goes from high concentration to low concentration. On the active transport, it can go from low concentration to high concentration. So does it require energy? For passive transport, the answer is no. Never. Doesn't require energy. It goes with the concentration gradient itself, so no need for extra energy to be put for each passenger side. An active transport, yes, it requires energy. Usually ATP to change the shape of a pump and allow a chemical to be squeezed from one side to the other. Okay, so do they require protein membranes? No, a membrane protein. And for passive transport, the answer is no if it's simple diffusion. And for facilitated diffusion, it requires a channel. For active transport, it's going to be requiring a protein pump, like the sodium-potassium pump. Okay, and examples for simple diffusion, you have oxygen and carbon dioxide gases that do simple diffusion. For facilitated diffusion, we have, for example, sodium channels in nerve cells. They go through facilitated diffusion through channels. And active transport, a good example, are good old sodium-potassium pump, 3 sodiums for 2 potassiums going from one side to the other. So I hope you had fun, and now you have time to do a quiz to check out to see how well you got the ideas of this class, and I'll see you in class. Cheers, bye-bye.